So for those of you who are less familiar or not familiar with Diagnostear, at Diagnostear we are focusing on developing and commercializing rapid point-of-care diagnostic solutions for ocular diseases, and we do that by biochemically analyzing the composition of the tear film. Our first commercial product is intended for assessment of dry eye disease. And I will not go into the technology. This is not the purpose of this session. I just want to say that this product is based on non-invasive, simple, and rapid collection of tear fluid using our proprietary microfluidic tear collector, multiparametric assessment or measurement of key tear proteins, and on-cloud smart algorithm for simple interpretation of the results. The first generation of this product is based on measurement of three key proteins in the tears. It has a sensitivity and specificity of over 86%, and it is approved for sale in Europe and in Israel. We have generated or developed our second generation or upgraded version of this product based on measurement of 5 key tear proteins. And recently we have completed a large longitudinal clinical study with LV Prasad Eye Institute in India. To the best of our knowledge, this is actually the largest and the most diverse clinical study ever performed on a diagnostic of dry eye. It involved 600 subjects, 500 dry eye patients, and 100 healthy controls. And we demonstrated that our second generation product is capable of detecting dry eye at all severity levels with 72% sensitivity, differentiating between severe and non-severe dry eye with over 80% sensitivity, detecting severe meibomian gland dysfunction, MGD, within the dry eye cohort with over 80% sensitivity, and finally, and most interestingly, predicting responsiveness to cyclosporin A therapy with 94% sensitivity, and more importantly, with a negative predictive value of 92%, meaning that we can pinpoint those that who will not respond positively for the drug. We thus conclude that pending additional validations and on a separate cohort of patients, our product may be used for diagnosis of dry eye at all severity levels and for offering proper selection of therapeutic alternatives based on the underlying etiology and the severity of the disease. Now, if some of you are going to attend the ARVO in Salt Lake City in May, Professor Sayan Basu, the principal investigator of the study, is going to present the full results of this work in the conference. Our second product, or second indication, is intended for differential diagnosis of red eye, namely for differential diagnosis of adenoviral conjunctivitis, herpetic keratitis, and allergic reaction. The product is primarily intended for general practitioners, for pediatricians, for ER, and for primary healthcare providers in general, but of course may be used by ophthalmologists. Results are qualitative, and the sampling of the tears is super easy, done by conventional, routinely used eye swabbing. We've been able, or we managed already, to design all the 3 separate tests and analytically validating them, and we integrated them into one simply used diagnostic device that we have designed and patented, and that was done with the great assistance of Nir Israeli and his exceptional team. Thank you, Nir. And these days we are putting all our efforts and our resources to clinically validate the performance of this product, and we do that on tears that we collect in few clinical studies with the names that you see here below. In 2025, we are planning to initiate external clinical trials for FDA and CE mark. Lastly, I want to say that in November 2024, Diagnostear completed fundraising and a merger, actually a reverse merger transaction with a Canadian company called Ocean View Technologies. And in December, the merged company called Diagnostear Technologies Incorporated started trading on the CSE, the Canadian Securities Exchange, under the symbol DTR. So now we are a publicly listed company. And finally, I want to thank all the team at Diagnostear for their hard work, and Israel Makov, Yaakov Miklin, and all the team at BioLight for their continuous support, especially during the last 18 months that have been extremely challenging for all of us. And thank you all for listening.
Good afternoon. My name is Oriah from Eye-Yon, the product manager of the EndoArt. Thank you for this opportunity to speak here. Today I will share the updates of our limited market release in Europe. So many of us know that there is a huge global shortage of corneas, but not only the shortage itself, there are many patients that are no longer candidates for human tissue because of multiple rejections. And the EndoArt is the first ever synthetic lamella to treat corneal edema. With more than 4 years follow-up and more than 400 implantations, we successfully improve vision and reduce pain for so many patients. So the key advantage of the EndoArt is that it is synthetic. It is available off-the-shelf product with 3 years shelf life now, non-rejectable, and you can see in the video that like this academic, it is attached to the posterior part of the cornea, but because it is a passive layer, it establishes and restores a new balance of liquids and nutrients in the cornea laterally. Here we present an interesting comparison between two groups of Eye-Yon cohort, EndoArt patients, normal patients and high risk. We call high risk patients, patients with multiple rejection, glaucoma tubes, ACIOLs, etc. We can see on the left that the CCT, the corneal thickness, reduced at the beginning and then remained stable in both groups. And also the vision outcomes improved on both groups. And now the EndoArt is positioned as a first-line treatment for high-risk patients. Over the past 1.5 years, we are in a limited market release in Europe. We focus on specific countries in Europe. We collaborate with top KOLs that can advocate for us in the podium, also through papers, and we choose specific sites with high volume of transplantation. We have an amazing number here of reorders, 90%. That means that every almost every doctor repeat ordering the EndoArt. And also we see that the sales, the adoption is the highest in Germany and UK, and it correlates with that the reimbursement and the regulatory processes were easy, not easy, but faster in those countries. So what did we learn from these 18 months of LMR? First of all, that reimbursement is crucial for adoption. The ideal EndoArt candidates are not the most complex eyes, not the worst of the worst, but also not the naive eyes, something in between. Keep collecting data, although it is very challenging in the real world. Stay close to the physicians and push to publish. Here there are examples of two articles published like this month and months ago of long-term follow-up, 2 years and 4.5 years, and we invite you to read them. And finally, the most exciting and important part of our job is helping patients. Our patients regain the ability to drive, read, use their phone, and this is an amazing story from the BBC. I would like to share it with you.
Harry carries out around 100 cornea transplants a year, but he only started performing artificial ones just before Christmas. With a human donation, there is as much as a 50% chance the body will reject it in the first 12 months. But that's not the case with an artificial one.
Because this is an inert material, it can't be rejected like conventional transplants. And therefore, this device is very suitable for patients who would be poor candidates for traditional transplant material.
Now, the actual artificial cornea comes from Israel, and it's described to be similar to a contact lens.
Day-to-day life is a lot better.
Today, he is back in for a checkup. Since the operation, there's been a significant improvement in his vision. What does that mean to you?
I was thinking that you might only end up with one eye. It's a huge thing. It's going to affect your life in many ways, like your job and driving and things like that. So to get the vision back, it's just so much better.
Thank you for listening. For more information, please reach out.
Well, good afternoon. It is my honor to present at the OIS, and thank you for your attention. Sanoculis developed proprietary stentless minimally invasive solutions for the treatment of glaucoma. Our mission is to bring innovative stentless technology platforms that can advance the standard of care and improve the outcomes for patients suffering from the glaucoma, all while leaving nothing behind. The MINT stands for minimally invasive nasal trabeculostomy. Is a mechanical microtrifine 0.5 long tool which creates a 150 micron in diameter trabeculostomy through the TM. And I'm happy to announce that MINT is being launched this year, and soon you will see the potential to the glaucoma patients. The MINT device stands for minimally invasive microsclerostomy, is the same concept, but here the trifine is going out 2 millimeter, in which creates a 90 micron in diameter sclerostomy in order to reduce the IOP. It can be performed supranasal or intranasal after failed surgeries. As you can see, the two products, MINT and MIMS, potentially cover the entire glaucoma population that requires surgery from mild to severe, MINT for trabeculostomy and MIMS for sclerostomy, using the same stentless concept of leaving nothing behind. Let's see a short video clip describing the MINT device. Our system is comprised of a foot switch, controller, and disposable handpiece unit, where the trifine is going out for 0.5 millimeter in order to create the opening into the TM. And it's a straightforward surgery. You prepare the eye, use gonio lens in order to visualize the TM. And here we create 5 openings through the TM to the Schlems canal in order to drain the aqueous humor from the These images represent an example of the safety aspect of the MINT tool, demonstrating its dimensions and perfect cut. You can see on the right the opening of 150 micron, and on the left how it looks like in human ex vivo tissue, where only the roof of the Schlems canal is removed. Here is another view through OCT images, the openings that we create into the TM. We conducted the MINT procedure as a standalone procedure on 66 patients under pre-market study protocol. The IOP was reduced by 35%, and the average number of medication was reduced by 76% after 1 year, and by 36 and 50 after 2 years. This slide provides a more detailed analysis of the mean 2 years follow-up. You can see that the IOP distribution indicate that the majority of the patient had 5 to 15 millimeter of mercury after the procedure, and in terms of medication, most of them had non-medication after the procedure. Using the FDA qualified success and complete success definitions, we can see clearly that the success of MINT is evident from the 2 years results. This gives glaucoma patients a promising, effective treatment. A short video clip just to show the differences between MINT and MIMS. So here, for MIMS, the trephine is going out 2 millimeter in order to create this sclerostomy. It's a bleb procedure. Physician prepared the eye, inject MMC under the conch, and introduce the device to the angle of the eye in order to create the sclerostomy. Again, this histology image represent an example of the safety aspect of the MIMS tool, demonstrating its dimension and its perfect cut. The image presented here demonstrate the drainage channel patency created by the MIMS after 1 year and 3 years post-op. It is still intact and perform its goal. You can see clearly how open and how effective it is. Sanoculis launched an FDA study for the MIMS device. The study design is prospective single-arm, 12 months follow-up in 129 patients, comparing MIMS standalone to trabeculectomy historical control of previous in-focus study. Let's hear short testimonials from 2 physicians who used the MIMS.
It was extremely intuitive. I think that's what I liked about it. For those of us who've done a lot of MIGs, not having to turn the head, not having to turn the scope, to be able to perform this procedure with a minimal small incision, a 1.1 and 1.2 incision, and to create minimal trauma to the target tissues without having to open up the conjunctiva, without having to worry about a foreign body left in the eye, that was a very pleasant experience.
This is a device that doesn't take up a lot of space in the operating room. One of the unique things is we don't actually have to use a prism to do the procedure, so we don't have to change the position of the microscope very much, which is a little bit different from most of the procedures that we're doing currently.
Sanoculis is in the process of developing a new addition to the MIMS toolbox that allows for a larger drainage channel diameter. As you can see, MIMS large, accompanying the MIMS and the MINT that we're about to launch. And I thank you for your attention. Thank you very much.
Thank you. Before I start, it's really an honor to be here in this community, and I think year after year to be here in the innovative community in this field is really outstanding, and it's an honor for us, and I'm happy to be here also specifically after I would say a tough year and a half. As Nahu mentioned, we're moving in a transition phase from an R&D company to a company that is on the verge of our first clinical study. And less than 5 months from now, we will be transplanting a 3D bioprinted cornea in a patient. And I'll just give a brief reminder of what we do and how we do it, and show the results, so the last steps before the clinical study, and hopefully next year when we present here, we'll be showing the clinical data. So Precise is I would say, bioconvergence company where we combine many technologies. We combine biomaterials, cell technology, bioengineering. We have a proprietary 3D printing technology that we developed, and a lot of engineering around that. And I'll show here about the quality control and how it is done. All of this has been developed in Precise. We currently have 3 products in the preclinical stage. We can see here at the top, this is the EK, the cornea product, which will be the first one into the clinic. Here on we see our retina product. This is a subretinal transplant. Here we're showing how we enter between the RPE layer and the photoreceptors and inserting the tissue. And here, at this bottom left one, we're intrastromal lenticule, also preclinical with over 450 days follow-up in the animal study. So these are the three products, and the results that we have are very promising in the preclinical stage. Here we can see the EK. Here we can see the tissue transplanted in the porcine eye. We have here a quiet eye, and at the bottom on the left, the intrastromal lenticule that stays clear and transparent, as I mentioned, over 450 days. What we have done over the last year, and this is essentially the new steps that we have done in the company, are moving into production. Moving a product like this into production is a significant challenge. And it has also the production itself, but also the quality control, and even more important than a regulatory path that approves this full process. And when we approached the FDA over 4.5 years ago and put us into the path that 3D printed tissues go, and said, that's a good idea, but we don't have it. And here, essentially, we built this path together with the FDA, and the full path from the cell expansion through the maturing of the cell, the scaffold, and the quality control processes that we have developed for each of these stages is fully agreed with the FDA, and based on that, we're moving forward, and we currently are doing the biofabrication in the Sheba Hospital GMP facility. And this is a step that we have done over the last year and a half. So that's step one toward the clinical studies planned in July. The second important step was the safety study, GLP safety study that we have done, where we show also efficacy and a very good correlation also to the pathology. These are preliminary results. It's important to say all of these studies have done at the biotech farm in collaboration with the Pharmaseed. And these results, essentially, we can see here the results where we have three groups, sham, scaffold only, and our product, which includes the scaffold and the human endothelial printed cells. And you can see here the results after 6 months. We're able to maintain the thickness and reduce the edema. And these results from the clinical aspect, from the efficacy, are very promising. They are directly correlated in what we see in the pathology. And also here you could see the three groups from left to right. The sham, where we also have a fibrosis, and also you can see the edema, which is marked here. The scaffold only, which I would like to notice here, the detachment of the scaffold. And our product here, where we can see that we have very good attachment of the product, clear and transparent, and the endothelial, the human endothelial cells are located here, and we can see it even more here in the larger image. So these results of the safety study are very, very promising. And based on this, we are moving, and we just this week submitted for the first study planned in July. The results, if we would just summarize them, so we see that there's no adverse reaction in the eyes in group 3, which is the product, and effectively we reduce the fibrosis and edema. Based on these two important steps, the manufacturing that we have completed and we have already done through 2 out of the 3 batches of production in the GMP facility, and based on these safety GLP results, we are moving forward toward the clinic. I must say that all of this is done due to our amazing team, and you just see the various disciplines that are involved, and this is what is really required to bring these tissues to the clinician's hands. So we have our biology team, biomaterials, physics, electronics, software, mechanics, engineering, QA, RA, and clinic. All of these come together to bring this tissue to the patient's eye.